Yeah. So today in this class we'll be seeing about database search tools. So now we will be seeing about BLAST, right? So what is BLAST? It is the expansion of BLAST is basic local alignment search tool. So as the word says, it depends upon the local alignment, right? So I have already said you what is local alignment. It will look for the highly similar regions in between your sequence also, right? So it will, and then why I have mentioned as database search tools, this particular tool BLAST, it will take your query sequence, or else you can say it as a target sequence. So it will take your query sequence and then it was going to compare with the sequences present in the database. So you have not only BLAST, you also have other database search tools, right? So in this class you will be looking about what is BLAST. So before going to that, why do we need to do the sequence similarity searches? Why we want to compare the sequences, your query sequence, with the sequences present in the database. So the sequence could be taken from your own lab, or else you can take it from your database, right? So that is called as a target sequence. So once consider that you have one sequences what you have done from your own lab. So here you will not know any informations of this particular new sequence which you got from your sequencing project. So okay, so when you don't have any kind of annotations, you can, with the help of your sequence, you can do lot of sequence analysis. Am I right? So one such analysis is to identify the similar sequence. Clear everyone? So in the process of annotation, you can infer lot of information for your new sequence. So when you do so, BLAST or database searching is one of the tool or one of the option to give you some kind of annotations for your new sequence. Okay, so you have some incomplete or a say without annotation, right? So you can take it from your or incorrect annotations. You can take any kind of sequence, and then for this new sequence, you can find the homologous sequences, and then you can also go for the construction of phylogenetic tree. So what is homologous sequence? They are the similar sequence which may have the common ancestor. Okay, so so in homologous also you have different kinds of sequences. Okay, like paralogs, orthologs, like that you have different kinds of homologous sequence also. So in your BLAST program, you can identify this homologous sequence, similar sequences. Clear everyone? So why database search? So to find out if you have a new DNA sequence, or else you can take it from your database, right? So this I have already said. So to identify the homologous protein to a putative coding ORF that may share a similar three-dimensional structure. Right? For these reasons also you can go for, you can identify the homologous, that is how far it is related between your query and to the sequence present in your database. All of you clear? So as I said, you can do the pairwise alignment. So pairwise means what? It is going, yeah, you can compare two sequences, right? So that two sequences can be sequence of your own interest taken from a database, or else sequence from your own lab. So that will be one sequence. What is the next sequence? It is the sequence taken from the database. So you have two sequences. So in BLAST it will do the pairwise alignment, right? So sequence 1, it is compared with the sequence 2, right? So this is an example showing you the global alignment and local alignment. See, in global alignment, it will look for the similar regions from one end to the entire sequence. It will just look for. Whereas if you take the local alignment, you can see the regions which are similar. See, for example, if you take this particular region, this seems to be similar to this region of your sequence 2 and here again. Right? So if you take this part of your sequence 1, it is similar to here or and then here again. So in this local alignment, it look for the highly similar regions in between your sequences. Right? So that is the difference between your global and local alignment. See, this is a slide which is explaining you the pairwise alignment search for your query sequence with the sequence present in the database. So here it is representing the sequence for what you are going to do the similarity search. So this sequence is called as a target sequence or as a query sequence. This sequence is compared with your sequences present in the database. Right? So you keep in mind that you are not going to take one database, you can take many databases also. So there is an option in BLAST where you can select one database or multiple database also. Okay, non-redundant database also you can select. So here in a database you will have lot of sequences. Am I right? So consider that this is a database. You have lots of sequences here which are represented in this image, right? So here this query sequence is compared with each and every sequence present in the database. So it is represented as black lines and then red lines. So what do you think about this? What does this red line indicates here? Yeah, very good. So they are the similar sequence to that of your query sequence. So these are the sequence present in the database. The sequence present in the database are having similarity with your query sequence. All of you understood the concept? Yeah. So now we will enter into the BLAST. So BLAST, as I said, it is basic local alignment search tool which is going to find the local similarity between your sequences, between the two sequences. So there are many different kinds of variants. Okay, so variants of BLAST are there, and we have different types of sequences also. What are the different types of sequence we can have? Yeah, you can have the protein sequence or else a DNA. So DNA sequence. So there is option in BLAST to compare a protein with protein, a protein with nucleotide, nucleotide with a nucleotide, nucleotide with protein. Like that, you have different kinds of comparison can be made. Okay, so for which we have different kinds, different variants of different flavors of BLAST is available. Based on your requirement, you can choose any kind of BLAST. Clear with this? Yeah. So this BLAST was developed by the scientist Atshul and his co-workers in the year of 1990. So can you say what algorithm is? I have said in the first class, no, in the few classes back, I have mentioned you what algorithm is adopted by this. Yeah, it's Smith-Waterman algorithm. So what is that algorithm? I mean, what type of alignment it takes? Local alignment. So for global alignment? Very good. It's your Needleman-Wunsch algorithm. Yeah. So we have some important terms what you have to remember in your BLAST. So one is your HSP. HSP it stands for high scoring segment pair. Can anyone of you say what is the any idea on this term? High scoring segment pairs. Why it is called as high scoring segment pairs? I wanted to explain you the scores, right? So when you will have more scores, yes, when you have more number of matches, right? So when you have more number of matches, obviously the score will be higher. It will be good, right? So you have the different segments in your BLAST, which will talk about your where it is similar, where it is dissimilar, where there is mismatch and all. It will you can get it from the alignment, right? So in high scoring segment pair, you will have the good number of matches. Okay, so that region will be having the highest score because the matches are higher. Clear? So those regions are called as high scoring segment pairs or else called as high scoring pairs. HSP or HSP. Okay, so this BLAST can produce several HSPs rather than a single aligned region. You can have the similarities, highly similar regions in between your different parts of your sequences. Where all it is similar, it will give you. So you will have the several short HSPs in your alignment, in your results. All of you clear with this? Right. So this is about the variance of BLAST. As I said, you have different sequences. You have the options to compare your sequence with different kinds of sequence. So to start with, we have nucleotide BLAST, which is named as BLAST N. So here it stands for, N stands for nucleotide. Okay, so nucleotide BLAST. What this BLAST will do, it will search a nucleotide database by using a nucleotide query. So the query is your nucleotide sequence. It could be your own sequence or sequence taken from the database. You take a sequence. To this sequence, it is going to compare with the nucleotide database. Can anyone of you see what is an example of a nucleotide database? In that, what database? GenBank. Okay, so in GenBank, it contains the nucleotide database. Right. So you take a query sequence as nucleotide, you can compare with the GenBank. Not only GenBank, you have other options also. Right. So BLAST P. So P stands for protein BLAST. Okay. So protein BLAST, what this will do? It will take your query as your protein sequence, and then it is going to compare with the sequences from your protein from your protein database. So give an example of a protein database. Very good. Your GenPET. Okay, in NCBI you have, and then here it will take UniProt and then PDB, right? So you have different protein database, right? So you have to give your query sequence, that is your protein sequence. So this BLAST P, it will, what it will do? It will take your query sequence, protein sequence, and then it is going to compare the sequences present in the protein database. Clear with this? Yeah. So next variant is BLAST X. So this BLAST, it will search your protein database using translated nucleotide query. So what you have to do, the input, the query sequence, it will be translated. Okay. So it will translate and then it will compare with the protein sequence. Okay. So T-BLAST-N, it will search the translated nucleotide database using protein query. So you have to give the protein query and then it will compare with the translated nucleotide database. So T-BLAST-X, it will search translated nucleotide database using the translated nucleotide query. Okay, so we have the different variants of BLAST. So based on your requirement, you can choose any kind of BLAST what you need. So we will see about nucleotide BLAST. So nucleotide BLAST, what it will do? And again, I forgot to say, in your protein BLAST, okay, BLAST P, you have the subdivisions like some different types of protein BLAST is also available, like side BLAST and fibroblast. Okay, so I will say that later. So nucleotide BLAST, it is going to search a nucleotide sequence with a nucleotide database, right? So it uses what algorithm? Smith-Waterman algorithm. Okay. So we will see the basic BLAST for searching your nucleotide sequence. So this will be the home page of BLAST. You can also go for genome BLAST. Right? So you can list all genomic BLAST database. You can take the sequence as a whole, as a genome, and then you can also go for genome BLAST. Right? So that is the option. So you have some basic BLAST like nucleotide. These are the different variants. So you have to, how to enter into BLAST is you go to Google, give the term as NCBI BLAST. Right? So you will have this page. So in this you have to select what type of, what variant of BLAST you need to. So now let us take this nucleotide BLAST. Right, you have to choose what kind of BLAST. So you will have a page where you can input your query sequence. So you can submit your query sequence, or else you can give the gene identification number, or else you can give the accession number. Accession number you can give, GIs you can give, or else your query sequence in FASTA format. So that is also possible. So here, apart from this, you can also upload your file. You can retrieve your, all of you know how to retrieve a sequence from a database. So you can retrieve and keep it in a notepad. You can browse it and upload your file. So that option is there. And then here there is an option to give the sub range also, right? Suppose let us consider that you have the sequence length of around 500, right? So instead of searching the entire length, entire 500 nucleotide, suppose if you are interested to domain a part, a domain region, let us take, in that you can give a sub range. I want to search from this particular region to this region. For example, I have to take from, so I can give us 150 to 300. So what this BLAST will do, it will go and take on, it will go and fetch only those sub range, and then it will compare with your sequences present in the database. Clear everyone? All of you? Yes. So you either you can take the entire sequence or else you can give the sub range. This is an option only. It doesn't mean that you have to give. You can give the FASTA format sequence or your accession numbers, and then you can search for the entire sequence. If you need, you can give the subrange. If your sequence is very large, and then if you don't want to compare entire sequence, if you are interested to view a part, so in that case you can give. Not always. And then you can give the job title. You can give any title of your job. So this is again an option only. So if you give, then you can have a very easy view, right? You can give the job title, and then there is an option called as database, right? So here you can choose what kind of sequence you want to compare. So for example, there is an option nucleotide collection. So this will take all the sequences, all the nucleotide databases which are present, and then it will compare. So you don't want to specifically choose. By default it will be. But if you want to choose one particular database, so you have to click this arrow mark, and then you can select any kind of database you need. Okay. And then there is an option to give a specific organism, which is again an optional only. So suppose if you are going to take a human sequence or else a rice sequence from the rice, and then if you want to compare it with only your model organism or to the rice itself, then here you can just give the organism name. Okay. So if suppose if you are not going to give here, it will search for all the organisms present in the database. Clear with me? Right. So this is an option. You can give specific organism. In that case, what it will do? The BLAST will go and search only to the sequences of that particular organism rather than searching all the organisms. Clear? So that is again an option. And then there is an option to exclude also. So you can exclude the uncultured or environmental sample sequences. So those sequences, if you are not interested, you can exclude. So that is again an option. And then finally you have to give the what type of BLAST algorithm you want to use. So in the down the page, you will have some other additional parameters. So by default, you will have some values here. Suppose if you are interested to change those values, you can give. So we have an general parameters like what is the target sequence. So here, based on the number given here, those many sequences will be displayed in your one page. So in the next page, you will have the next. See, for example, here it has mentioned as 100. So here it will give you the maximum number of aligned sequence to display. It is going to compare with so many sequences present in the database, right? So in that it is going to pick the sequences from highly similar sequence to the very less similarity. So that is how it is going to give in your display page, in your result page. So if you give the target sequence as 100, what it will do? It will fetch you the 100 maximum results in your first page. So if you give 50, what will happen now? 50 sequences will be displayed in the first page, and then next 50 will be there. Yeah. So next 50 will be displayed. So likewise, you can give your maximum target sequences to be viewed. All of you clear with this? Yes, anyone is not clear? Right? Everyone are clear. So here it will mention you what is the maximum number of sequences which are similar to your query sequence to be displayed in your output page. So, and then whether short queries, whether automatically we have to adjust the parameters if you are going to give a very short input sequence, whether the tool has to adjust with this different parameters based on your short sequences, right? So that is again you have, if you check, then automatically it will adjust. So here you have the options like threshold. So for example, if you give as 10, so it will take the at least to this threshold level, it will look for the similarities. Okay, so to at least to 10 residues, it will just have the what are the possibility of having this particular matches. Okay, at least to 10 it will take. Birth size. So here the blast, it will look for the number of words to be matched. Right? So suppose if you give us 3, then it will search for at least 3 residues. 3 it will take. So here if you give us 10, it will look for the word size to be 10 residues to be matched with your sequences. Right? So this is again an option you can leave as a default value also. And then you can also search for these options also, matches or mismatch score. See, as I said, all of you know what is gaps, right? So what is gap? What is gap? For getting an optimal alignment, you are giving the gaps so that you will have the best matches between your sequences. So we are inserting gaps in your algorithm. That's why we give the penalty score, right? So this penalty scores can be matched. Okay, so gap cost or matches and mismatches. See, for example, match and mismatch score. For matches, it has taken the score to be 1, and for mismatch it is 2, and for gaps you can give the score. Right? So you can leave this as default value, or else if you need you can change those scores. Right? And then you have the options to filters for filtering or masking. In the sense, you have some kind of regions called as a low complexity regions. So in nucleotide or in some sequences, you will have as a continuous NNN. So that has unambiguous sequences. Okay, so this, if you leave as such, it may affect your, it may have some effect in your similarity searches. So you can exclude or filter those regions, or else you can mask your repeat regions also in case of your proteins, right? So, one protein may have the repeats, or else in your nucleotide you will have some repeat region. So those repeat regions can be masked. Okay, so you can filter those regions. So these are some of the options what you can change, and finally you can give us a blast here. Right? So now you have the result page. Listen all of you carefully. So you have the result page. So here it is displaying you the 3 different kinds of display. Okay, so in this page, in the result page, the first part you will have a header information. So in header information, you will have what is the query, what sequence you have submitted, what is the description of it, what kind of sequence it is. So if it is nucleotide means it will mention, or else if it is a protein, it will, here it will mention as a protein sequence. What is the query length? Okay, the sequence what you have submitted, no, what is the length of that? What are the databases it has selected? See, for example, it has given as NR, means non-redundant database. So here we are not chosen one particular database. So that's why it is comparing all the sequence database. So for nucleotide, it has compared with GenBank, EMBL, DDBJ, and other database. Right? So suppose instead of NR, suppose if I choose GenBank, now what will happen? Here database, instead of NR, it will be mentioned as GenBank, because we have selected an option to look for the similarity only with one particular database. Right? So if you are chosen like that, it will be compared only with one database, or else with the all the database. Clear? Right. So and then what kind of program you are using? This is the version and this is blasting. So this information you will have in the header information. And next one is your graphics, right? So in the graphics, you will have the barcodes. Okay, so here it will represent with the color codes, right? So starting from red to black, it will be displayed. So what does this colors indicate is that if it is going to be in red color, they are highly similar regions. Okay, so after that you will have pink, green, blue, and if it is going to have a very, the very less similarity will be given in the black region, okay, in the black color, right? So here what does these red color indicate? So the each and every line or the bar, it represents the sequence what it is compared. Okay, so what is the alignment? It is represented in your in your graphical output, right? So if you click here, it will be taken to the alignment part of this similarity search, right? So it is taken the sequence, your query sequence, and then it is comparing with the sequence present in the database. Am I right? So for each similarity, you will have as a one color which are represented in one line. So you can, if you click that, you will be taken to the alignment part. Clear with this? Right. So here, can you see some regions here? Yes, can you see? Here it is red, here it is pink, here can you see green color? So those are the regions which is not very highly similar. So this belongs to this category. Right? So here from the color code you can indicate, you can identify how well it is similar, whether it is highly similar or moderately or very less similarity. Right? So now down the page you will have the description of your this, everything will be one after the other. So in the first page you will have the header, and in the top of your page you will have the header information, and then followed by the graphical, and then you will have the one line description, right? So in one line description it will take the, what is this, any idea? Yeah, accession number only. What is this representing? Very good. So it has taken a query sequence and then compared with the sequence from the database. So this is the first sequence what it is compared, right? Your query sequence is compared with this sequence taken from the database. That description is given here. What is the score? Here it is not representing the alignment. It is just giving you the description, what sequence it is taken, what are the scores, what is the query coverage, everything I will explain now. So it will give you the maximum score, it will give you the total score, and then what is the query coverage, what is the e-value, and then what is the maximum identity. Okay, identity percentage it will give you. So maximum score, it is the highest score, or else it is called as a bit score. So this score is calculated by summation of all your matches, mismatches, and your gaps. So when the score is higher, obviously it means that you have the highly similar sequences. Okay, so when the score is higher, you can say that it is similar regions, similar sequence. So you will have the similar total score also. Next you will have the query coverage. So query coverage is also very important. So what is this, what this part explain you is, what is the percentage of your query covered with the sequence taken from your database. Okay, so here 100% it is covered, right? So say for example, in your sequences, you consider that you have the sequence length of 500. So this 500 sequence, it can be compared with the different regions present in your database, sequences present in the database. So what is the query percentage? Okay, how well your query sequence is covered with the sequences when it is going to do the alignment. So 100% it is covered. See here it is having only 73%. What does it mean? So 73% of your query is, the similarity is found with your sequence present in the database. So how this value should be? Higher or lower? Higher. So when it is higher, then it means that major part of your query is being covered. Okay, and then your e-value. E-value is your expected value. right? So this expected value, you remember that it should be less. So what is this expected value explain is that what is the you got an alignment, right? You have got an alignment. So to this alignment, it will give you the score called as E value. So you please remember that E stands for expected value, that should always be less. So what does mean is you got some alignment. What is the chance of getting your, what is the expected chance of getting the better alignment than what you got, right? So that should always be less. So you got for an optimal alignment, the E value will be always less. Clear all of you? Everyone? Yes. So E stands for the expected value. What is the number of alignment expected by chance? Okay. So if you're going to have the higher score, then you will say that you may have some expected for having a better alignment than this. Okay. So when the E value is lesser, it means that the alignment what you got is good. Okay. And then down the page, finally you will have the alignment. Right? So in alignment, what is the sequence? It has aligned. What is the database? Your description of the sequence, what it is compared, it is given here. So you will have the scores, what I explained you, all your bit score value, E value. What is the percentage of identity? Okay, how much identity? See, for example, 2789 out of 2789, 100% it is having the identity. Okay, so if you have, for example, 1000 means what? Suppose out of 2789, you may have, when you go down, you will have that. Okay, because it is taking sequences from the database. So to what percentage, what is the identity between your sequence to the sequence compared with your database, it is mentioning here. So 100 means what? 100% it is identical sequence. And then gaps, how many gaps are there? 0, because it is highly similar, right? So 0 gaps are here. So it has used the plus strand. So this scores will be given here, and then you will have the query. And then what is this subject sequence? Query is the sequence you have taken for comparison. So it starts with 1. What is this 1 represent? This is your first residue, first nucleotide, and then it is going to 60, and then this is 60 of your query. This is your 61. So what is the subject sequence? Anyone? Yeah, very good. It is the sequence taken from the database. That is your subject sequence. So subject sequence 1 of your query is matched with the subject sequence, first nucleotide of your subject sequence. So like that it is comparing. Since it is entirely it is similar, everything is being similar. See sir, you have the lines over here. Can you see? In between your query and your subject, you have one line. Right? So these lines represent that they are matched. Okay, matching residues. Suppose if you don't have match, so these lines won't be seen. Since it is present, it means that these two nucleotides are matching. Okay.